heralded a new time in science. Before microscopes, scientists believed in a theory called the theory of spontaneous generation. According to the theory of spontaneous generation, living things randomly, spontaneously developed or arose from dead matter. Scientists left dead things or decaying things such as steak or a piece of meat. They would leave it outside, and out of it would come maggots. And so the idea was it's likely that these living things just spontaneously just came out of nowhere. But then some scientists did great work. There was a scientist called Anton van Leeuwenhoek. There was another one called Louis Pasteur. Alongside other scientists, they did lots of great work, and they invented the microscope. The microscope allowed us to see a world that had never been seen before. Probably the most diverse world, called the world of microscopy, microbes. So they took a sample of that meat and they put it on a microscope, underneath the microscope, and they saw tiny little cells that gave rise to the maggots. So the theory of spontaneous generation was disproven. Now, as we disproved the theory of spontaneous generation, it gave birth to what we call the cell theory. Cell theory states that all living things come from pre-existing cells. Number two, all living things are made up of cells. Number three, the cell is the basic unit of life. These three tenets make up the cell theory, thanks to the microscope. So let's talk about a microscope. I have here one of the microscopes we have in lab. I took a picture of it, and it has some important parts that I want us to know. This is a light microscope, okay? It's a light microscope. It uses light, just refraction of light. It uses light for us to be able to view what's present underneath the microscope. This microscope has some important parts I want you to know. Let's start here with the ocular lens. The ocular lens of most microscopes that we have here are going to be 10x. That's the power. That's what you look through. You look through the ocular lens. Then you have here the objective lens. The objective lens is what directly looks at the object you're viewing. So your eyes go to the ocular lens. The objective lens looks at the object you're looking at. In most microscopes, we have 3 or 4 objective lenses. The first one, the 4x lens, is called the scanning objective. It's used to look. It's broad. It's bigger, and it's used to look at a wider surface area. So you scan for the object you're looking for. The 10x is the next one up. This is called the low power objective. Next up is the 40x, which is called the high power objective. Some microscopes have 100x, which is called oil immersion. For our lab, we're going to use these 3. Okay? The ocular lens multiplied by the objective lens gives what we call the magnification of a specimen. As a scientist, every time you use a microscope, please write down the magnification. A few other parts that I want you to know here on a microscope. We've got the stage, where the sample goes to. And the sample can be clipped here by a stage clip. The stage can be raised and lowered by the coarse focus knob. And then this allows you to see better what you're looking at. And when you finally see what you're looking at, you can fine-tune it using the fine focus knob. Okay, here we've got a light source. Chapter 3 talks about the cell. Our focus is the animal cell, which is also congruent to the human cell. And we're going to try to draw what a generic cell looks like. I have great expectations that everyone here will be able to draw a cell. Okay? Please do know this for the sake of your exam also. Okay, the cell, let's start with the outside of it, the cell membrane. This is also known as the plasma membrane. It has many functions. It's made up of lipids, and it functions as a barrier to the outside world. It's important in signaling, which means if there's going to be communication with the inside of a cell, the communication usually starts with the cell membrane. It's excuse me. It creates a transport barrier also. It's used for transportation of things inside and outside a cell. And there are a few more functions of the cell membrane. Please look up functions of the cell membrane. Okay, the plasma membrane encompasses the inside of the cell, which is known as the cytoplasm. The cytoplasm contains a liquid called the cytosol, and that contains everything on the inside of the cell. There is a giant part of the cell known as the nucleus. The nucleus separates us eukaryotes from prokaryotes. The nucleus has pores known as the nuclear pore. That's the nuclear membrane, and then the nuclear pore. There's a region in the nucleus that's really dark, known as a nucleolus, and it's the site for ribosome formation. So the nucleus, the main function is to carry DNA, right? Deoxyribonucleic acid, which is our hereditary material. It carries our genes and also directs the manufacture of proteins. Okay. Continuous with the nuclear membrane is what we call the endoplasmic reticulum. It's a network that's found within the cell that's useful in synthesizing proteins. The endoplasmic reticulum there is one that's rough, known as the rough endoplasmic reticulum. It's rough because it has ribosomes on it. That's the rough endoplasmic reticulum. It has ribosomes on it. What are ribosomes? I'm going to indicate ribosomes in blue. Ribosomes are the site for protein synthesis. So new proteins are made at ribosomes. The ribosomes that are free, that are just floating free of the endoplasmic reticulum, are known for synthesizing proteins that will be used inside the cell. Whereas those on the endoplasmic reticulum are used to synthesize proteins that are destined for outside the cell. That's the rough endoplasmic reticulum. Okay, for instance, a protein like insulin would be made in the rough endoplasmic reticulum because it's produced in the pancreas but shipped outside the cell to the bloodstream. So we have another type of reticulum known as the smooth endoplasmic reticulum. The smooth ER, as it's otherwise known, SER, is a site for lipid synthesis. In some structures, in some types of cells, it's also the site for calcium storage. Then we have another structure known as the Golgi apparatus. Golgi. The Golgi apparatus is the site for packaging and shipping. It packages proteins that have been synthesized, and then it ships them outside the cell. It also does what we call post-translational modification. Post-translational modification means that after proteins are made in the endoplasmic reticulum, the Golgi has the ability to add other things to these proteins like glucose, and it makes it more suited for whatever function that protein has. And when we get to the endocrine system, we'll talk about this. We have a few more things within the cell I want you to know about. There is the powerhouse of the cell, which is called the mitochondria. It's where ATP is made. There is what we call the lysosome. The lysosome contains enzymes that digest old organelles, and sometimes when the lysosome erupts, it digests the entire cell. Then we have what we call the peroxisome, and as the name implies, contains peroxide. The function of the peroxisome is to detoxify. It removes toxins and free radicals from the cell. We have what we call the centriole. The centriole is responsible for manufacture of spindle fibers. I'm going to talk about that briefly. The function of spindle fibers is to aid in cell division. And then we have, I'm going to use yellow to illustrate cytoskeleton, which is basically the skeleton of the cell. So those are the major things I want you to know about the structure and function of the cell. Every cell in the body, except for certain cells, most cells will undergo what we call mitosis. There are some cells that don't divide, and there are some cells that are sex cells. But some cells, especially in the structure known as the brain, may not divide, but have other ways of increasing capacity. Mitosis refers to nuclear division. That's division of the nucleus, specifically the nucleus. So which means mitosis can happen even if a cell doesn't divide. There are some cells that have lots of nuclei. Can you guess anyone that has lots of nuclei? Muscle cells. Muscles can have multi-nucleus or multinucleated. Okay, the first step in cell division is called interphase. Interphase is the stage where a cell is not dividing. Okay, a cell is not dividing, and you have the nucleus and you have what we call chromatin. Chromatin is just chromosome jumbled up together. That's interphase. Our cells spend about 23 hours of 24 in interphase. The next step is then mitosis. Mitosis has 4 stages. The first stage is prophase, and this is when the cell gets ready to divide. The nuclear envelope begins to disappear. The chromatin condenses and forms 2 sister chromatids, known as chromosomes. And then also during prophase, the centriole migrate to the poles in readiness for cell division, and they begin to make what we call spindle fibers, which are going to aid in cell division. The next stage is metaphase. During metaphase, spindle fibers are formed completely. The spindle fibers are formed completely, and they attach to the sister chromatids. So this is metaphase. Usually during metaphase, the the chromosomes will align in the equator, the center of the cell. Then we have anaphase. During anaphase, the sister chromatids are pulled apart. This is called anaphase. An event called cytoskeleton may begin to happen during anaphase, and this is when the actual cell begins to divide. So remember, mitosis is division of the nucleus, but the division of the cell is called cytokinesis. So we've got interphase, prophase, metaphase, anaphase. Next up is telophase, and I'm going to fuse telophase and cytokinesis. So cytokinesis is division of the cytoplasm, and you can see the cytoplasm is beginning to divide. Okay, and then you have your daughter cells already forming. This is mitosis. All right, there are lots of questions on mitosis on our quiz. Please take time to answer those. There's a concept called diffusion. And diffusion is the movement of solute particles, or particles in general, from a region of high concentration to a region of low concentration. So for instance, if I spray a perfume, the perfume is going to move from a region of high concentration, which is where I sprayed the perfume, to a region of low concentration, which is your nose or anywhere else that's not the original place where I sprayed the perfume. So diffusion allows particles to move. If this is a cup and I put a bunch of particles on this side of the cup, diffusion means that the particle will move around until there is balance within the cup. That's diffusion. Diffusion is very important in our everyday lives because it's how our cells feed. For instance, my hands are distant from my stomach, but all the nutrients come to my stomach. So the way my hands get fed is because food finds a way to go from my alimentary canal to my hands, to my face, to my feet, to different parts of my body. This is called diffusion. A specific type of diffusion, known as osmosis, is common within the body. Osmosis is the movement of water. Usually, movement of water from a region of high solute concentration to a region of low solute concentration. So if I have a cup and I divide it into two, sorry, that's not a cup, and I put a membrane in between like that, this solute is present in water. Okay? Two things can happen. The solute can move, but sometimes that's difficult because some solutes are large. So what happens often is water moves. If water moves, it's going to move from a region of high solute concentration to a region of low solute concentration. I'll give you a quick illustration. When I was a kid, I had a fish. That's not how to draw a fish. I had a fish. It was an electric catfish, which means it would shock you whenever it's in water. So what happened is the fish came from the ocean, and for some reason we put it in tap water. A few days later, the fish died. The science behind why the fish died is something I only understood when I got older. I got slightly older, I'm like, oh, now I know why the fish died. Ocean water contains lots of salts. So the fish itself is used to high saline environments. And then we have tap water all around the fish. And that is a low solute environment. What happened with my fish was that the tap water began to try to balance by entering into the fish. Eventually the scales of the fish started to fall off, the fish started to swell, and the fish died. This is an example of how water moves from a region of low solute concentration like here to a region of high solute concentration like here. Okay. The solution where the fish is found is called a hypotonic solution because it has low solute concentration. If this was the inside the fish, will be a hypertonic condition. And that's because there is more solute in the fish than in the environment. I hope that makes sense.